Welcome to our presentation on how the microbiota may or may not influence cardiovascular disease development. We'll begin our presentation with the learning objectives. We have 5, which are listed here. The first is to evaluate how the microbiota may influence factors affecting cardiovascular disease. Second is to provide a mechanistic link between microbial metabolism of nutrients and the development of atherosclerosis. The third is to be aware of the lab and diagnostic methods used to monitor and establish the prognosis of cardiovascular disease and related symptoms. The fourth is to assess how diet and exercise influence gut microbiota composition and diversity. And finally, to provide some practical recommendations on probiotics or prebiotics that may positively affect the gut microbiome and their role for lowering cardiovascular disease risk. We'll move on now to the introduction and a little bit of background on the issue. So cardiovascular disease is number 1 at killing us. According to the CDC, over 600,000 people will die every year from CVD. That's 1 in every 4 deaths, a huge number. 735,000 people suffer heart attacks every year, and these numbers only reflect cases within the United States. So, show me the money. The economic impact of CVD is absolutely staggering. According to a policy statement recently released by the American Heart Association, in 2010, the direct cost of heart disease was estimated to be $273 billion, with indirect costs of $172 billion, and these numbers are projected to increase by the year 2030 to just about $1 trillion collectively, and that's an increase of 61%, or to put it in perspective, 1/18 of the current United States national debt. As a side note, nearly 40% of the U.S. population is expected to have some form of cardiovascular disease by the year 2030. So that's just crazy. So we would want to know, as physicians or physicians in training, what is causing heart disease and what can we do about it? So the research so far has only shown about 20% of CVD is attributable to some direct genetic cause, so we start thinking about alternatives, most obviously environmental influences on CVD, and a fairly obvious one is the diet, food as medicine. So why the diet? We know just intrinsically and surrounded by information everywhere that certain diets are better for us, but we don't really know everything about why that is. Why is it that a 108-year-old woman who's eaten bacon for her entire life has no cardiovascular disease, and yet somebody might die of a heart attack at the age of 60 with a similar diet? We don't understand. So the concept of microbiota is relatively new. How does the microbiome interact with the food we eat? And what are the consequences for cardiovascular disease?
So now we're going to talk a little bit more about how the food we take in is digested and metabolized by the specific commensal microbes in our gut, and how that could potentially provide a mechanistic link to the development of cardiovascular disease. So current research has shown specifically that our intake of dietary phosphatidylcholine has led to the development of different metabolites that we'll talk about that can lead to atherogenic plaques in our blood vessels. So this is groundbreaking work that has shown how our gut flora can significantly influence how the dietary constituents that we take in are in our body, and how it may influence the metabolism. So you can see by the diagram on the right that when we take in dietary phosphatidylcholine from different sources such as meat, fish, milk, and eggs, it's acted on by phospholipase D that converts it into choline. And then once this choline gets transported into the gastrointestinal lumen, specifically in the colon, the commensal microbiota will convert that, specific commensal microbiota will convert that choline to trimethylamine, which is subsequently transported and absorbed into the blood and is converted by hepatic flavin-containing monooxygenases into trimethylamine oxide. And that is the specific compound of interest of many of the research studies, TMAO. And in addition, choline and betaine are produced through this same metabolic pathway. And these 3 metabolites, TMAO, choline, and betaine, are of specific interest for promoting atherogenesis in blood vessels. More specifically, 4 different organisms that are shown to undergo this type of metabolism are Clostridia, Proteus, Shigella, and Aerobacter, which are present in the colon and can convert choline to trimethylamine oxide. So how specifically does TMAO cause atherosclerotic development in our blood vessels? This has also been a research topic that has been recently explored. So dietary choline and elevated serum levels of TMAO have been associated with enhanced macrophage accumulation of cholesterol and also have been shown to form foam cells, which are vital for formation of atherogenic plaques. In addition, other research projects have shown that TMAO will stimulate scavenger receptors to also promote these macrophages to uptake lipoproteins and cholesterol. So these are two key factors that lead to the development of atherogenic plaques. In addition, it was found that the hepatic flavin-containing monooxygenase 3, FMO3, is necessary for this production of TMAO in humans. So only those bacteria that contain FMO3 will be able to convert the choline into TMAO to cause this atherogenic process. In addition, they found that TMAO in the presence of aerobic growth conditions may cause inhibition of electron transport chain in the mitochondria, may lead to oxidant stress to cause detrimental effects in the cells, and can cause mitochondrial uncoupling.
Okay, so let's look at diagnostics for cardiovascular disease. So is it possible that we have a new biomarker for heart disease? Well, several studies that came out of the lab of Dr. Stanley Hazen and his colleagues at Cleveland Clinic have demonstrated that elevated plasma levels of choline and its associated precursors and TMAO are linked to an increased risk of CVD and poorer clinical prognosis. Interestingly, these results are independent of traditional risk factors for CVD such as age, blood pressure, smoking habits, diabetes, and other prognostic markers such as C-reactive protein and myeloperoxidase. So we're just going to take a quick overview of the studies that led to these findings and then see if it's possible that this could be used clinically in a routine manner. So one of the earlier studies had about 1,800 patients that were followed for 3 years, and their levels of choline, betaine, TMAO were measured, and it was found that there was a dose-dependent relationship with the increased risk in CVD in phenotypes such as peripheral artery disease, coronary artery disease, and myocardial infarction. These relationships were preserved after adjusting for heart disease risk factors, medications, and renal function. These findings were validated in a larger cohort of about 4,000 patients. And again, they found that TMAO levels are associated with increased risk of major cardiac events. Interestingly, this is also true for patients who are low risk, so those who are younger than 65, those who don't smoke, and are generally healthier than those who are in the high risk group. They did another study, again, looking at TMAO levels and precursor levels at the same time. And what they found basically was that it's possible that you could measure the precursors such as choline, betaine, and TMAO, but the prognostic value of these precursors is only valid when TMAO is also elevated. And that might make sense because TMAO is the product of the metabolism of these precursors. So, as you can see here in the graph, that holds true. And lastly, since we're talking about cardiovascular disease, this group also looked at heart failure. So patients with heart failure had higher levels of TMAO and increased long-term mortality than those without. And so measuring TMAO levels in this patient population might have good prognostic value as well. So overall, a major limitation is that the studies all came out of one institution, but they provide some of the best insight into these disease mechanisms. And so, in fact, this group now is on their way to developing a commercial product in partnership with Procter & Gamble. that one day you may see at the drugstore for patients to help manage their levels of TMAO. Additionally, the Cleveland Clinic is also developing a routine test that can be used to measure TMAO. So it's possible that we may see this as part of a routine panel in the future. So the major question is, what will you do in practice with these test results? How will you treat someone who has high levels of TMAO? And that has yet to be determined.
Diet contributes significantly to the microbial populations of the gut, as well as their metabolic activity. When microbiota homeostasis is disturbed, dysbiosis leads to disease. Dietary alterations can be responsible for 57% of the variation of the gut microbiota population, and changes in diets can affect microbiota populations within 24 hours. There are three main microbial genera or enterotypes: bacteroids, Prevotella, and Ruminococcus. Bacteroids enterotypes tend to be more prevalent in diets high in animal protein and saturated fats, for example, Western diets, while Prevotella enterotypes are linked to diets high in carbohydrates and simple sugars, for example, vegetarian or vegan diets. As Mark mentioned before, dietary L-carnitine that come from animal products can be converted to TMAO and lead to increased cardiovascular risk. This conversion from dietary L-carnitine to TMAO did not occur in long-term vegetarians or vegans who have been vegetarians or vegans for over a year. And furthermore, vegetarians, vegans not only have lower plasma concentrations of TMAO than omnivores, but also produce less TMAO. This suggests converting dietary L-carnitine to TMAO is a microbial adaptive response due to this consumption of red meat. Physical activity also plays a significant role in modulating gut microbiota, although most evidence is based off of rodent models. Exercise rodents had more diverse microbiota composition and diversity than in sedentary rodents. Due to the limited research on humans, it is unclear to what extent gut microbiota composition is affected by exercise, independent of diet.
So, are there any pragmatic recommendations today? Manipulation of the microbiota. This is a new field of promising research. The focus so far has been centered on measurable changes in cholesterol levels. It involves eating live bacteria cultures that are traditionally found in yogurt, cheese, and fermented foods, of which include lactobacillus, lactococcus, and bifidobacterium. These cultures possess an enzyme, bile salt hydrolase, that has been shown to reduce cholesterol levels in animal studies. But why focus on cholesterol levels? For one, it is an easy marker that is measured. Secondly, there is a 1 to 2 rule, where a 1% reduction in total cholesterol levels equates with a 2% reduction in cardiovascular risk. To place these numbers into context, the power of a 1% cardiovascular disease risk reduction, if sustained for 10 years, has been estimated to prevent 25,000 new cases of cardiovascular disease, prevent 3,500 deaths, and save around $46 million per year. Returning to the bacterial cultures, to understand the importance of bile salt hydrolase, or BSH, it is important to recap where bile comes from and its connection with cholesterol. Bile salts are formed from cholesterol. They circulate in the gut and are reabsorbed in an ongoing cycle. The enzyme bile salt hydrolase of the bacteria cuts the water-loving molecules that makes it easy to reabsorb from the gut. Instead of being reabsorbed, it's secreted in the feces. So, is it time to start shopping or cultivating at home? Well, not so fast. These studies have had an inconclusive range of results. Total cholesterol levels and LDL levels either did not change or slightly decreased. More strikingly, these short study lengths, small sample sizes, and lack of standardization have been insufficient to generate significant conclusions. However, the future is ripe. Any questions?